All right, we started the chapter 1 last week. So briefly talk about, what the causing the infectious diseases in terms of we call that pathogens, if you remember that. Infectious disease by the pathogens. So this is actually the key word that we learned last week too. Every single thing we talk about pathogen in terms of that bacteria can be a bacteria. Bacteria can be a pathogens, viruses. Oh, actually, I need to divide it into two. Pathogen can be divided into two, which is a micro or the macro, the levels of that too. In terms of micropathogens, I can divide it also categorize into the cellular or non-cellular, or the living or non-livings too. This is well summarized on your page, the first page of this. Page 2, I'll call the cellular and the non-cellular. Now, perfect example of the cellular, microcellular pathogen is, as you know, the some bacteria or fungi could be the examples too. Oh, actually, I actually had to do this again too. In the cellular and non-cellular, we can be prokaryotic or the eukaryotic. Prokaryotic, typical example, bacteria. Eukaryotic, typical example, fungi is a good examples too. You may include the protozoans in the eukaryote as well too. Non-cellular micropathogens can be included in the things like the proteins itself too, protein code, the viruses. But it's a typical example of that all the times too. Macro is pretty simple, so you can think about living inside the body or outside the body too. Those living inside the body is known as the ecto. Living inside is endo, and that's ecto. So you can think about ticks, mites, all that kind of included in here too. And we went through some of the details about the pathogens in the plant, pathogens in the animals too. That was what we learned last week in terms of giving some introduction of what the pathogens are, and we went through many of those. And the homework was all about determining the different type of pathogens and all that too. So no problem. But to be honest, you won't get the questions especially each of those two. Rather, you actually get a question related to the agricultures, or maybe related more into the plants, or even for the animals later on too. That's something we're going to discuss in the second half of the class. We also talked about the chain of the infection, if you remember that too. Described it as there's infectious agent, reservoir, and the port of the exit, mode of transmission, port of the entry, and susceptible, the host, which is kind of giving us the why we have to do that in terms of the prevention, how to stop the cycles too. That's what we actually learned last week as well too. So it was really useful things we talked about. Today, the first half of the class, we're going to talk about then the pathogens again, but more about historical background of this. How would people know that all of those can cause the infectious diseases too? Infectious diseases all the time too. Now, so to understand that, we have to think about what was the theory before these two. The theory that using by infectious disease by the pathogens is actually known as the germ theory. Germ theory. And this is by it's one of the famous the scientists all the time, Pasteur. Pasteur. He's a French. The Pasteur is the one that proposing the germ theory, which is basically that we have as pathogen, most likely the microorganisms are causing the infectious diseases. Prior to this, before the germ theory, there was actually a theory called that spontaneous generations. Okay, there was a theory called the spontaneous. Write it down. Spontaneous. The generation theory prior to that germ theory. So it might be good to give me some ideas that it was basically pre to the 19th century. Germ theory was published known from that in 1800, so from germ theory in 1860 or 1861, around that year. So what was the spontaneous degeneration theory? This believed that the living organisms like include the include the microscopes, microbes could emerge from the non-living substance. So, example, what's this? Rotting meat are living organisms could like spontaneously produced the maggots as one example. And another thing is the dirty rags that are generate the mites, which is causing the pest at the times too. Okay, and then they believe that all the diseases arose from what they called as the miasma, basically known as the bad air. Okay, rather than the microscope. Rather than microbes. Rather than the microbes. So, if you ever remember, like, so what happened to the Middle Age of the Europes? Have you seen like the people wearing the kind of weird mask of the kind of bush shape of this? Okay, they believe that. So you have seen that from like some, like I don't know, horror movies and stuff too. The people will actually believe that the bad air can cause the diseases too. So they have some ideas about infectious disease, but it was basically thinking about there was something that non-living organism can cause the diseases too. This was something that disproved by many scientists, including the Pasteurs, and this is something we're going to talk about today. So let's think about how this spontaneous generation theory, gets disproved by the some experiment or scientific experiment. Page 43, please, in your workbook. It shows that one of the very famous experiments called as Swan Neck Flask Experiment. Swan Neck Flask Experiment. It's one of the very important, one of the most important experiments in the biology and the microscope all the times too. Now, what is this? This is basically like how this performed too. For this question, okay, on your workbook, question 5, page 48, please. You can copy this one down, and what I do is I'll give you like some of the things that we're doing in a scientific experiment. So let's start from the aim of the experiments first. Okay, aim of the Swan Neck Flask experiment. This is by the Pasteurs. What Louis Pasteur is to determine the whether the microorganisms arise spontaneously, okay, or come from the environment. So, looking at the aim, this is actually in the testing for the spontaneous generation theory, because they believe that living organisms could emerge from the long substance all the time, so spontaneously. In other words, it doesn't matter whether we have microbes or not too. If we have anything that is a non-living organism, like a rotting meat, they always produces that of the maggot, regardless of the environment situations too. Okay, take one. Okay, one for you. So, in terms of that, Pasteur put the hypothesis for this. Now, what he hypothesized in here is the microorganisms do not arise spontaneously. Pasteur didn't really believe that microorganisms, or the called the pathogens, cannot be just arise by themselves. Instead, okay, Jennifer, this is a page 48. You can copy this down here. Instead, they come from the pre-existing the microbes, okay, in the air. So for this, he put it up to some of the independent and so variables and the dependent and control variables for that too. Okay, you can refer back into the diagram on your page 43. This is a swan neck experiment by the Pasteurs too. But independent variable, he actually put it into the shape of the flask. So one is basically this swan neck versus the open flask. Dependent variables. Dependent would it be like the microbe growth? Okay, it would be like the measurement. So you're just gonna see the whether the microbes, okay, or microbial, or I should say microbial growth. Okay, because in bone growth. Okay, control variables, running out of space. Control variables. You use the same type of the broth. You use the bone broth in the case of this. Duration of the duration of the boiling, and also the exposure to the air. Okay, temperature he used, all controls too, and obviously the environment conditions, all that. Okay, so page on your workbook, page 43 describes the method as well as the apparatus and what he actually used to. So as you can see that he prepared the three flask. Okay, one with the swan neck, all the swan necks too. What he does first, he put the broth into the swan neck, the flask, and then he kind of sealed the end of that too. And then what he does, well, he applied the heat to kill, like, just in case, like kind of sanitizing the effects too. Then what happened? One of those got left as a left flask just sit like this too. Okay, one of those flasks we open the neck of that too, kind of breaking the top of that. Another one is we hit apply the flask is sideways and the let it sit as well as too. So there'll be a slight bit like an air exposure given that too. This was his method. He boiled the broth in the swan neck flask to kill the microbes. Let one flask stay intact and the broken neck of another flask, and observe the micro growth in the over time too. The result was quite interesting. What he Pasteur found that was the both of those flasks, which is one with the open neck, one with the seat, I guess in the sideways too, had a bacteria present or microbial growth. But one that has the heat applied and the seat as just sits just horizontally too, there's no bacteria growth. So this is kind of telling us that both intact. So let's talk about the result here. Something that's very important. Broth in the swan neck in intact to the swan neck, the flask, okay, and the remained, we use a term called sterols, sterols. I start right, ILE, so. Okay, and broth in the broken, the neck, or the open flask. Okay, show the microbial growth. This was the result, that one with the exposure to the air had a bit of like a microbial growth. One without the exposure to the air didn't have the they sterile. Basically, there's nothing really going to. Now, let's talk about the conclusion of this. This is a highly valid, in terms of validity, reliability, accuracy, this is a highly valid, highly reliable, highly accurate, because Pasteur performed the experiment for many times to perform that too. Conclusions is a very important thing. We have to write it down. So conclusion from that result. Pasteur concluded that microbes, they do not arise spontaneously. Okay? Instead, the microbes, okay, come from the pre-existing the microbes in the air. In other words, this is disproof the spontaneous the generation theory, spontaneous theory, and supporting something known as the germ theory of the pathogens now. So we're going to think about why is it kind of disproving this, first of all, given here too. According to the spontaneous generation theory, if this is true, if spontaneous generation theory is true, what happens to this swan neck experiment, if that is true? All three of them, including the one with the closed flasks too. Because according to spontaneous theory, it basically says that as long as we have a broth, which is the non-living organism, non-living things, it will produce their germs, it produce the microbes spontaneously. It will do that. But Pasteur proved that, hey, we have a broth. If this theory is true, we have to have like some bacterial growth in here. But as we just put it in here and seal that flask, there's nothing happen. It was the first time like disproving that, kind of scientific methods too, proving that this is a new theory coming out. We call the germ theory, but the Pasteur is coming too. The importance of this Pasteur the experiment is it was a new era. People don't need to wear this kind of weird shaped mask anymore too. Instead, they are trying to think about what are the germs, how do we prevent the diseases, how do we cure the diseases after that too. And after that, this research about the germs was exponentially growth. Yes?
Can you explain how the swan neck flask actually works?
Swan neck flask is how it actually works.
Doesn't air go into the flask?
No, we basically seal that. You seal that too. Now, as you seal the whole thing, after we stir the whole like a broth there too, there's no microbes because they would reach into certain temperatures too. And Pasteur was actually an expert of the killing the microbes. He was actually known as the pastoralism or the vaccinations, father of that too. So he knew that by heating up some of the broth, it would kill the existing microbes. There's no microbes living in here. This would have some air from the air. The microbe will go into the broth growth, the microbial growth. Here, because it's a swan neck, kind of S shape, there could be a chance of the air is not like heating is not enough to kill the microbes in the existing this kind of shape of it there too. So, chance of that we have the bacteria surviving here, there'll be a minimal, but there's a chance, which is proved by there is a bacteria present after that too. Does it make sense to all too? So this is known as the Pasteur's experiment, which is a very important theory I'm generated from, which is called the germ theory by Pasteur too. you can copy this spontaneous generation theory down on your question 3, page 46 as well too. You can copy this one or germ theory given that too. So there was a question 3 given. So what is a germ theory? Basically, germ theory is the one we discussed the last week, that pathogens is causing the infectious diseases all the times. Or pathogen as a germ, as you may think about. Germ theory, let's write it down as a definition of germ theory here now. Germ theory of of disease is the correct term to use. It states that microorganisms, whether they're bacteria, viruses, are the primary causes of the infectious diseases. So very simple. There's something we know. We learned it. Pathogens basically causing the infectious disease. But ideas are just generated from Pasteur by doing something, a scientific experiment like this, kind of using the aim, hypothesis, variables, all the kind of structures that we followed too. Okay, previously it was given that spontaneous generation theory. You can copy this down as I explained it before too. Does it make sense? Okay. So on your page 45, the previous question 2. I give you some of the summary of what the Louis Pasteur did in the given times too. I give you some of the narration of what he has done in the experiment and the purpose of that, all given. Everyone good? Yep. Okay. So, give you some the timeline of how this germ theory got developed, and therefore coming to the Koch's postulate later on too. Let's start with the Pasteur's experiments. He did quite a lot of experiments. So the experiment. First of all, here was done. Experiment 1 was a fermentation experiment, and that held in around 1857 too. So mid 1800s. Now, this proved that microorganisms, in case of that yeast, okay, are responsible for the fermentations, okay, but not the spontaneous generation. So Pasteur now from this experiment had some ideas about, hey, we have microorganisms, they are doing something about the fermentations. So he now developed that into the famous swan neck, the flask experiment. That's held in 1861, after 4 years, around the next year too. As I explained that before too, this is very important, as this is completely disproof the spontaneous generation theory. Generation, and what it does, it was support, known as the germ theory. Now, germ theory was pre-existed before the Pasteur as well too. There are scientists actually proposing that claim that okay, there must be germs causing the infectious diseases. But this is the experiment that proving this is the right theory, we still follows that too. After that, he actually developed the idea about this germ theory and invented, or started, something known as pasteurization. And this is something that we're using for the milk even nowadays too, in 1864. how does it do? We are basically, it's kill the harmful like microbes in the in liquids. Okay, and without the changing taste. Basically, this is well used in milk productions. Did you guys know, like in the raw milk from the cow, we can't really drink it because it contains a lot of the bacteria, to harmful germs too. So to deliver the milk in your grocery stores, what we do is we put the milk in the glass jar and put it into kind of water bath, but in the kind of low temperature. Because if you put in the high temperature, what happened to the milk is it's kind of separating the oils, which is the butter part and the cheesy parts too. We don't want to go up to like changing the taste, the formation of the milk itself too. So we are lower the temperature, but it's enough to kill all the harmful microbes in the milk. This is why we are able to drink the fresh milk. And if you think about the expiry date for the milk we're getting into a grocery store, it's fairly long enough. Maybe go for a week or two. Okay, due to that, as we're understanding that some importance of the temperature as well as sterilize or sanitations too. And most importantly, after that, he was involved in vaccine development. And that happened in around 1880s. Okay, so what he has done, he developed the vaccines using the weakened or the dead, the microbes for, now we known as the immunization. This is Pasteur's experiment or his achievement, started from the fermentation all the way to the vaccine developments too. Okay, we are talking a little more about the vaccine development in chapter 3 and chapter 4 later on too. But vaccine development in terms of that, we talked about transgenic organisms too, or or how to change the DNAs. But understanding that, we are now able to know that this is it possible to prevent the infectious diseases, as we understanding that infectious diseases is caused by the pathogen or the microbes or the microorganisms. Case of this. Okay. Any questions about Pasteur's experiment? Any understand, like any things, do I need to explain it again, of the Pasteur's achievement of that too? Okay. Most important thing would have been the swan neck flask experiment, but basically pasteurization is an application of that, the experiment as well too. All right. Now, along with that Pasteur, there's another the scientist who actually supporting the germ theory as well too, known as Robert Koch. So, on the same page, could you write it down? His postulates too. So we're going to talk about Robert the Koch, and I would say his experiments too. And the different subheadings too. So Robert Koch, he published a paper and published ideas about known as Koch's postulate. Postulates are powerful, some ideas which is kind of proven by many scientific experiments too, in 1876 to 1884. So if you think about the years, this is after the Pasteur's in the swan neck experiment. So he already knew that the germ theory, he was going to follow into germ theory as well too. Then, what he actually proposed from that is he established, using his postulate, he was able to establish the steps to prove the microbes. The microbes cause the diseases. We're going to talk a little more about this in a few minutes. Okay, so he actually wasn't more going deep into the very specific type of diseases too. One was the anthrax. Okay, and he was doing the experiment with that. It was famous, known at the time, killing a lot of people in the Europe, and he proved that this anthrax is caused by the bacteria called the bacillus, the anthracis, anthracis. It causes the anthrax. So he proved that this is actually a germ that causing the disease. Okay. A few years after, he also proved that tuberculosis, TUBR, or simply known as TB, tuberculosis. Okay, discovery, or shortened known as TB, was 1882. Okay, and he proved that this is he proves the mycobacterium. So MYCO, mycobacterium. The tuberculosis. Okay, as the cause of the TB, the disease, which is a very serious respiratory and respiratory infections, can cause many people too. And later on, he also discovered that cholera. Okay, identifications. In the year of 1884. And this is basically, he identified called the Vibrio or Vibrio cholera, CHOLERA or cholerae, yep, as the cause of the cholera. And most importantly, and prove that any like a waterborne transmissions. So two scientists, Pasteur and Koch, contributed to a lot of things that we know as the germ theory. Even nowadays we're using that too. There's no better theory than germ theory. Even now, they're proving that infectious disease is caused by the pathogens. Prior to that, there has a spontaneous generation. Also, prior to the spontaneous generation, they actually thought that is cause of the curse of the religious regions. So you probably haven't seen like in something like the Bible saying like maybe people get sick because of the curse of someone too. Nowadays, we are proving that it's not because of the because of the curse, but it's more likely because of the microorganism nowadays too. Okay. So write this one down. Although two scientists and their achievements given in terms of that what they have done to develop the germ theory. So page 44. Let's go back to the question 1. Sorry about the going here and there. I'll give you some summary of that what each of the scientists done and how they contributed to the our understanding of the pathogens or the infectious diseases now. So you can make a table of the scientists. So let's think about the Louis. Started with him. Louis Pasteur again. Yes.
What's the word
Before.
Like the last.
Identify Vibrio cholera as the cause of the cholera and to prove the waterborne transmissions. Okay. So let's talk about his discoveries. Just give you a summary of that. The discoveries and the importance of that too. If you don't have time to revise that, this would be a good table so you can use that too. So, what has he done? He first developed the germ theory of the disease. Okay, and he by the famous swan neck the flask experiment. So it is very important to know his setups, his variables, hypothesis, methods, as well as the result and the validity of that too. And it can be summarized that he studied the pasteurizations, killing the harmful germs in the milk or like some of the foods we consume. And he also developed the vaccines for rabies and anthrax. So why is it very important? The importance is he revolutionized the microbiology. So from then, since then, we now understand about how to study the microbes. But before that, we didn't really know the microbes in the management details too. And he led to the sterilization. Always get like confused about the spelling. So sterilization. Okay, all the techniques as well too, which you may see in the medical dramas, what they do with that, the sterilizations too. Basically, if you want to sterilize, we are using the alcohol swab, we boil that, get all the microbes that exist too. Okay, and then he also is the one that the paved way, for the vaccination. The vaccination, like a development too. This is his achievement or the importance of the Louis Pasteur's discoveries. Okay, let's talk about a bit of Koch then. Robert Koch. He kind of discovered or established that he established Koch's postulate. Okay, and as I gave you that too, he identified the tuberculosis, tuberculosis, and anthrax the bacteria. Okay, and then he also proved many, but very specific. Proved the specific, the microbes that cause diseases. So why is this very important? The reason why he could do mainly identify many of the microbes because he had very good, established scientific methods. Scientific methods for the disease identification. Okay, this means that it is improved for the doctors, especially like the medical practitioners, the diagnostic. Okay, and later on, it influenced the antibiotic, the development, as well as epidemiology. So these two are the fathers of the microbiologists too. For since then, we're able to study for the microbes, which is not really visible in the naked eyes. Okay, so let's see what has Koch and what experiment he has done and how he made the postulates too. Page 41 of your workbook, please. This is the experiment known as Koch's experiment, which is like the backbone, the basic, like helping you to postulate, make a postulate of his own postulate about germ theory. So it's all well described in here, basically. So what he has done was this. Koch collected two samples. One is the healthy organisms, two in disease organisms. From the healthy organism, he took some of the blood and put it into microbes, and basically there's nothing really shown. So it was basically known as the controlled experiment. This whole thing, it would be the controlled, the experiment. What that meant is he was able to use the microscope to see like actually possible causative agents too. Obviously, in healthy organism, there's nothing to be observed that too. So he put it at his controlled experiment first of all. Then he collected a second sample with the disease and the died organisms, like a rat, for example, given. What he has done, he took the sample out of the blood sample out, and then he put the maybe like a suspected agent from that too. Put it into that, cultivating the agar plate after we're taking that out of the samples. Okay, and the cultivating that too, and injecting that sample, the cultivated sample, into the healthy organisms or healthy rats. What he has found out that soon after, the rat died in the same cause or the same disease type. He took the on the blood sample out, and we basically found that whether we have the same causative agent presented or not too. I know it sounds really like common sense nowadays. And nowadays it's kind of common sense because obviously if you suspect that the rat died because of the infectious disease, you do not touch it because you know there could be germs in there. But in the age of the 1800s, they didn't have any ideas about that yet. Because they believe in this, remember, the spontaneous generation. They believe that living organisms would not causing that disease. It's probably something else too. The rats probably like died because of the the microbes, but not from the air. Probably something else too. So based on these experiments, the Koch's experiment, he was able to postulate the four very important. So this is going on your question 4, please. Page 47. Let's talk about Koch's postulate in summary of that given. Also known as Koch's postulate. So under the title of these, he had four points of that. 1, the suspected pathogens, causative microbes must be present in all diseased individuals, okay, but absent in the healthy individuals. Two, the pathogen must be isolated, okay, and grown in the pure culture. So I'm talking about the agar plate in the case of this too. 3, when the cultured microbes okay, microbe is induced, or sorry, too much physics, introduced into a healthy, the organisms or the host, okay, it must cause the same disease. Now, from there, the pathogen must be isolated or the re-isolated, okay, from the newly infected host. Okay, and must be identified, okay, as the same original microbes. This is a Koch's postulate, basically describing what the experiment he has done. Okay, so why this is very important? Why these postulates are very important? Why this whole experiment set is something key point to understand the pathogens, and we're still following that ideas even now too. Okay, importance of this. So you got to think about importance of this postulate. One, it is one that provides the very scientific, which is scientific proof of the that microbes cause the disease. In other words, it is supporting the germ theory. Okay, and also it was enabled the identification of the bacteria or the bacterial, especially bacterial, the pathogens. Okay, and this is kind of giving him that the TB as well as anthrax identification related to that too. As I said, this is basically further developed too, so improvements in the disease, the diagnosis. Still we're using the pathologies in the lab a lot of times too, but not only that, using the pharmaceutical treatments as well as the preventions. So this idea was basically related to the vaccines later on too, and the antibiotics developments as well too. So from these ideas, it saved millions of the lives actually. Think about the vaccines or the treatments that we have for the coronaviruses too. Actually, ideas can start from this whole Koch's postulates too. But it has one limitations. And the limitation related to this second postulate, pathogen must be isolated and grown in the pure culture. When I say the pure culture there, this is actually, I'm talking about, like, for example, agar plate. Having some agar gels with the nutrients in it. Okay, but, Aaron, have a think in this way. Can viruses live in the agar plate?
Yes.
With the nutrients only?
Can?
They can, but can they reproduce themselves in the agar plate?
Not really.
Not really. So, agar plate is specifically designed for the bacterial growth. Okay, the nutrients, kind of, think about sugar basically. It only has sugar and some of the proteins inside. Viruses doesn't need that. They need instead the host cell. They need you. Because if you think about the reproduction of the bacteria compared to the bacteria to the viruses too, bacteria need some nutrients to reproduce themselves, whether there's a binary fission, asexual reproductions. But viruses need a cell, host cell. So they get invaded into host cells, spreading their DNAs and all the genetic materials into producing themselves a lot too. They cannot produce by themselves without the host cell. So Koch's postulate was fantastic, except it was only limited to the bacterial growth. Okay, so you got to think about the viruses. It's a limitation. And there's something that we have to talk about later on too, is viruses requires require out a living host, the cells to reproduce. Means that following to the postulate, in making impossible to isolate and the culture in the agar plate. We'll culture them as bacteria. Does that make sense? So there was remain as one of the limitations of Koch. Okay, so basically also related here. But despite the limitation of the viruses too, now we realize that viruses are another key feature, like the pathogen can cause many of the diseases too. Back into that, a lot of the bacterial growth will be kind of cool too. At the same time, have you guys heard of penicillin? Penicillin, like basically used the same time, at similar times, the penicillin was kind of invented, kind of found, discovered, and invented too, which can save a lot of millions of soldiers' life from the infection due to the bacteria. But viruses was not actually discovered at the time yet, because as you know, the size of viruses are in kind of micro, like in nano levels, compared to the bacterians too. So even using the most developed microscope at the time, they couldn't actually see the viruses. We realized that sometimes after, the viruses can cause the infectious diseases too. Okay, so these are the summaries of the question 1, 2, 3, 4. Did I pretty much answer everything? 1, 2, 3, 4, 5. I did 5. I did 4. I did 3, 2, and 1. That 2. Giving you some ideas about the germ theory of by the Koch as well as the Pasteur's too. It's a really long story, but I try to make it short. Try to give you a lot of summaries using the tables, all the experiments too. But something that's very important to understand. Try to understand it as the timeline of that too, how this get developed, and it's giving you some better ideas to understand and memorize it at the end too. Any questions for these? Fairly interesting part. Okay. All right. What we're going to do, we'll take a little break because the next part is going to be a little intensive in terms of the disease and agricultures too. So have some break, have some drink, and we're coming back to the second part then. In your worksheet that I just gave you, could you find the page 11 and page 12? It gives you some of the cause and effect of diseases in agriculture. So we talk about some specific examples today. Okay. All right, you probably have an experience of seeing that white mold in the plant look like this too. This is also a bit infectious disease. It's basically by the pathogen type of the fungi, and that causes a stem rot in many plant species, including the beans and canola, and they have the light brown soft patches and the cotton ball-like growth. Look like some of the eggs of the bugs too, but the stem causing the rot and the plant dies eventually too. So what we're going to talk from now is we're going to talk a little bit about some type of the infectious disease in the agriculture, including the plants like this. On the plants, potato leaf rolls too, given that the virus type. In case of that, infectious disease causes the stunt growth, low growth, and the smaller of the field potatoes are produced from the plant. Potato plants will die if the infection is severe enough. What I recommend is at least memorize 2 examples of the plants in agriculture, 2 examples of the animals or the livestock in the agriculture sport. Just get prepared. There was one time that questions were given as narrate or describe the disease, infectious disease in agriculture and how to prevent them too. I recommend to memorize these 2 for the plant. For the livestock, you can memorize these 2. There's something related here. Now, this is the livestock. We can have the trichomonolysis which are basically parasite of cattle, cause the sexually transmitted infections in the cattle and disease spread during the mating or through the contaminated equipment used during the artificial inseminations. And infected animals can be difficult to identify as there are few visible symptoms. Infected cows will have the fertility issues. They may abort the calves, have the difficulty getting pregnant, or be unable to become pregnant at all too. That's one. Or we may think about the also known as the major health and safety issue for the farm workers, also be had to carry the bacteria symptoms, or also known as the another type of that in your workbook. I put a lot of that too, but given the case study, page 54. This is quite common in the sheep or the cattle too. So, foot rot in the sheep. Could be a little disgusting, but it's basically what the cattle's infectious disease went through. So, foot rot is an infectious disease of the hooves of the sheep's, goats, or the cattle caused by the pathogenic bacterium. This Colobacterium nodosus. It causes a painful abscesses between the toes, lamina, and the weight loss and gazing is affected. So, let's talk about what could be the possible factors of that too. So, because as you know, if we can identify the factors, we're able to prevent it, and possibly able to eliminate it too. Most of those livestock living in having that foot rot is one reason they are living in the environment of the very moist and the muddy. The conditions. This is one of the prime factors of the having those to be bacterial get growth, because wet and muddy and damp environments means what? They are favorable conditions for the bacterial the growth. In terms of the moist, a lot of water is available. Muddy means that there will be nutrients available too. Damp environment, that means that they are increasing, basically, a lot of the increasing the infection risk. Especially if they get a cut. Now, have a think about that in here. Even though they're living in the muddy and moist, there's something we cannot control it too. But when the time that there is an overcrowding of the livestock, this is the times that close contact between them, close contact between the animals, that will allows this fast spread, the rapid spread of the bacteria. Okay, a lot of times this will now followed by the poor hoof, okay, and hygienes, and also be a care. I mean, like farmers, think about the farmers in Australia too, they get really fairly big too, so they don't really consider, they couldn't consider about one livestock having the disease. So it means that the trimming of the cleaning creates conditions too, so lack of the trimming and the cleaning of the creates conditions. It's basically creates a more bacterial growth as opposed to. Okay, there's a chance of that. Use of the contaminated. Okay, contaminated. The pastures or the equipment used as well too. So bacteria can leave that, in case of that, can stays. Bacteria can survive, or they can just remained in the tools or the equipments used in the farms too. This, if you know like cause of that, it's easy to be cured too. Basically, the injection or something like you're using the antibiotics or some of the antibiotics, like some topical like some creams can cause can cure that quite easily too. That's one of the things we can think about the food treatment ships in terms of that. Now, plant disease are a little more like significant too, time to time, because they are more overcrowded. So next page, page 55, please. We have in Australia a lot of the plants grown, kind of blessed country. We have grains, fruits, and the vegetables, folders, fiber, horticulture like plants, and the forestry plants, the blood plants being used in the creations too. All that. Now, this is really free of the damaging plants too. But in terms of that, you probably have seen the leaves with the black dots. This is due to the fungi kind of causing that. Or sometimes we're seeing that a plant with the insect, the mites are grown to, or even bacteria can cause that the damage to fruits or the vegetables can sometimes of that too. Let's then, well, he's giving a lot of examples, viruses, kind of giving all that. I just try to put the amount of pictures and all that too. But page 62. I want to focus a little more on to what the factors that contribute to the outbreak of the infectious diseases in agriculture entrepreneurs and basically related to Australian plants as well too. A lot of times, so let's make a table for this again. Question 1. Let's go for the possible factors that cause that infectious disease in agriculture. 1, as we talked about overcrowding in the livestock too, high population. High populations on the density would be considered to be one of the prime factors of the spreading the germs and therefore spreading the infectious diseases too. So in terms of that, you can think about crowded conditions. Okay, that will facilitate the rapid spread of the pathogens, okay, among the animals and all the plants. Example. So explanations here. Explanation. And the example. Examples of that, as we discussed that too, it could be a foot, any of the foot, or the mouth disease. Mouth disease. In kettles. Okay, and basically it could be a viral infections. Can cause that a lot of times too. Another factor that can contribute to the rapid spread of the disease would be the poor biosecurity. Biosecurity like measures. So what do I mean by that? Basically this, lack of the quarantine. If we know like one of the stock gets like affected or the plant, we have to like separate, isolate the two. Sometimes we have to eliminate the factors. Or lack of quarantine could be possible lesions too, or improper. The sanitations could be another factors as too. Okay, and could be the movement of the infected. The livestock from the farm to another farms too, or even for the plants from the market to somewhere, and that will increase the transmission significantly too. So, have you guys heard about it's nowadays really hard to buy eggs in the grocery stores too? Egg prices going in the skyrocketing highs too. Or like you have a limit to buy how many eggs. Now that's actually due to that, the bird flu or influenza. So avian influenza okay, in the poultry. And basically they are spread through the feeds and the waters a lot of times too. Or the bird flu can be considered to be here. Okay, and guys, don't just think like, oh, okay, we have a minimal effect on this happened in the farms too. No, we actually get this affected in our grocery stores. So influenza, sorry, the influenza is how we spell it. Okay, could be happens. What else? We could have the possible environmental conditions too, in case of that. So what about the environmentals? As I said about the moist and the muddy conditions too, some of the conditions like they are favorable conditions, okay, such as the humidity and the humidity, could be like temperature, could also be a soil quality. Okay, can promote the pathogens and pathogen survival, and also be spread as well too. It could be also related to the vectors like mosquitoes or flies, which is a macro like a parasite, or like you can think about how they grow in what kind of environment they favor as well too. So in terms of the examples too, I give you one example in the plant. The late blight is a type of the disease. Late blight, RLBRGHT, in potatoes. Okay, and they basically thrive the cool, wet condition. They thrives in the cool, also be wet conditions. Okay, type of the bacteria can grow in the plant as well too. So try to give it 3 examples, the 3 factors, and how they can affect the like agriculture too. Now, I won't spend too much time on that, to be honest, too, because you only got one question type. Whether like asking you describing the one of the plant or the livestock disease in the Australian agricultures. We can prepare them as we go, but these are the main things about the factors that increase the spread of the disease in the agriculture, the farms too. Yep, there are more vectors: climate change, biodiversity loss, habitat fragments too, globalization and trade. Now, you can also cause the infectious diseases. Did you know that? Okay, if you're visiting the farm, and sometimes like the farms requires you to have it like a sanitizing yourselves too, because you can carry the germs from the external environments too. There are many other factors, but something that I'm not an expert of the, to be honest, agriculture in here. I'm an expert of science, more likely to. So it's good to know what are the difference between them. But let's talk a little more about the pathogens. What are we talking about? Bacteria, viruses, prions, protozoa, all the microbes. Okay, you got to understand that they are able to mutate. In other words, they are able to adapt to some different situations too. Do you remember what we learned in Module 3 in terms of that there are variation in the population? We call the genetic pools. Okay, there will be selective factors or selective pressure happens, and there will only be a few selected, called the natural selections too. Scary thing is that can be happen in the world of pathogens as well too. Whether that's welcome or not, it's actually happening too. In other words, we got to actually understand about possible adaptations. Page 64, please. Adaptation of the pathogen to facilitate their transfer as well too. They are able to evolve, they're able to adapt to the different changes. Sometimes there's a reason why using the excess amount of the antibiotics will actually strengthening the bacteria so that we have no antibiotics or the medication to use. I give you a lot of things in more details in the workbook too, but what I do here is let's answer the question in pretty much the last page. Page 73. I give you a summary of how pathogen can adapt to facilitate infectious diseases too. If you have a kind of depth study to do, use my workbook ideas to I can write a demo ideas, making the better answer. But let's summarize some of how the pathogen can adapt first of all. Now, the way the pathogens, ways of pathogens adapt in terms of that, there are 6 ways we can think about in here. Pathogen, first, they can evade or evading the immune system. They kind of develop somehow, whether that be adhesions or making them stronger too. 2, they can also multiplying and spreading really efficiently. They're able to adapting to the environmental conditions too. Four, they can developing the antimicrobial, including the antibiotics, resistance. They can also adapt or evolve to exploiting the host factors and also be the niches. 6, they are able to promoting the transmission. These are the 6 ways of the pathogen can adapt to invade your body, invade anything like anything that given to. So let's be a little specific about how each of the microbes, I'm talking about bacteria, viruses, protozoans, fungi, and prions can adapt, or can in the examples too. On the same page, same diagram here. Let's talk about some of the pathogen types first of all too. And how do they actually adaptation in summaries given. So, started with bacteria. How do they adapt? Think about the property of the bacteria, guys. This is the prokaryotic, unicellular, whole lot of 2, means that they are able to reproduce themselves really quickly. So, the rapid reproduction, and from that, possibly, and the mutation is the strength of the bacteria. For the, I need more space, so for antibiotic resistance, that's one. In other words, they are able to biofilm formations, and also they can produce toxin production. They can produce some toxins, and scary thing is they go through something known as horizontal, the gene transfer. And that's a possible for the prokaryotic bacteria too, unlike the unicellular, unicellular too. Horizontal gene transfer means they can transfer genes from one to another really quickly too, in the case of they're in this selective pressure. Yes?
Did we learn that in Module 5?
Did we do that?
Horizontal gene transfer.
Not really. Kinda.
I don't know. When I was studying for it, I was
Did
like,
you write it
what is
down?
this? What is this?
But basically, I'm talking about the same thing, like bacteria, like in the Module 5, the genes and genetics too. And that's related to the, if you really think about it, the CRISPR as well too. Okay, viruses. How do they So that this could be really more scary too. Viruses, if you think about how they reproduce themselves, they need a host cell injecting their DNA out of the genetic materials in. Therefore, they have really high mutation rate, really fast, really rapid. And this is the reason why you took the 3 or 4 boost shot for the COVID-19 coronavirus. Okay, so what they do, they integrate integration into host cells. Okay, because like integrated into host cells. Again, and using, I love this term, using the host machinery for the replication, for the reproduction or the replication, I should say. This means that a lot of things that are given that to, they are antigenic like a variation will be given to. Now think about here. From one host cell, there will be, let's assume like there are tens of viruses can be reformed to. Among them, there's a good chance of the mutations happen. So it's not just talking about one mutation, there could be various mutations can be happen. So even now too, we believe that there are probably like hundreds of the mutations of the coronavirus from the 2019s. Protozoa is another type of that too. Now, let's think about protozoa too. Okay, protozoa, Hayden, is this the prokaryotic, eukaryotic? Pro? What's a protozoa? What's a protozoa? Not prions. I can tell you, prions are the misfolded proteins, which can cause like mad dog disease and stuff. We learned it last week in that could be a really worst case scenario. If you get a mad dog disease, the fatality will be 100% basically. Protozoa, Aaron. Unicellular, multicellular, prokaryotic, non-cellular, cellular. Unicellular? Yeah? Unicellular? So cellular or non-cellular? Protozoa. Non-cellular. It's a protozoa is? No, protozoa is a cellular. Oh, yeah. Yep. Eukaryotic, prokaryotic. It's eukaryotic, so it's like a fungi. So they're kind of in the same family as fungi too. So protozoa is the following kind of similar method of the reproduce themselves like a fungi too. So they are having like really complex the life cycles with the multiple hosts. Now, what they do is their ability to, there could be like, again, antigenic, the variation. It can cause that they change the genetic structure of the cells or the nucleus, and also be, it has ability to form the cyst for the survival. What about fungi? Let's continue to talk about those now related. They are able to tissue, something like tissue like a digesting enzymes. Digesting, they can produce those enzymes easily too. Okay, and important thing is there could be antifungal resistance with something known as the biofilms. Simply just thinking about they can create their own barrier, the bio barrier, to protect it from themselves from the antifungal. You can think about medications too, or it could be like topical creams we put it onto. So sometimes you probably realize that cold sore is a good example. Okay, if you have a cold sore, that's a fungal. Now, you probably realize that cold sore can be spread from here to there a lot of times too, if you don't put the topical creams. But sometimes, even after you put the topical creams, they will not really goes away really quickly too. because fungi can actually kind of spread and can just build it up, adapted to like a given changes to. They're building up some barriers called as a biofilms too. So it would take a little more time to kill all that, eradicate all that too. Sometimes it would not be effective. In case of that, you need to switch the type of the creams you put it on. Okay, that's also related to the type of those two. Prions is basically, okay, one thing, let's finish with the prions. Have a space, sorry, running out of space as you write it here. Prions, as you know, they are misfolded. They're known as the misfolded the proteins. Okay, and they're quite scary because they can be resistant to heat and disinfectant. And they can have the long incubation periods, because they're simply a protein themselves too. This means that they don't really need the nutrients. They don't really need anything host cells to. They can exist as just the protein by themselves. So if you're unlucky, ingest the protein, misfolded proteins, you can cause a severe damage to your organs as well as all the tissues too. Okay, and they're able to be resistant to the heat because they're simply a protein by themselves. This is how the prions can be adapted to the changes. But unlike the prions, other things here, especially bacteria, the way we can eradicate the bacteria is simply sanitizing, basically boiling, using the boil water at certain temperatures too. aka we can cook them, we can kill them too. So this is why we preferably using the at least 60 degrees Celsius to wash your the utensils, the kitchen things too, or even using the hospitals a lot of times. Okay, using the alcohol or microbial things too. But think about it. If we have some superbugs, superbacteria, which can be heat resistant due to the formation of the microfilms too, means that those bacteria can survive in even the high temperature. That could be scary because especially in the hospitals too, the people will not be no longer be safe to be from the bacteria too. Yep.
For bacteria.
Yep.
Horizontal gene transfer.
Mm-hmm.
So is that when the bacteria gets like a mutation and it's a favorable trait and they can transfer it or?
Kinda. So that's one very unique property of the bacteria, which is a prokaryotic, and they can go for the asexual reproduction quite rapidly too. Okay, so basically this is why they have the plasmid though. Plasmid is something they like circular DNAs we're using for transgenics too. Plasmid is actually known as I describe the kind of piggy bank they carry that too. In the case of that, they need to replace the genes. They took that from the plasmid. They have that too. Or some other structures. So they're easy to amend the damaged DNA structures too, means that they're easy to get adapted to the given changes. So compared to the antibiotics that we used to use 100 years ago, nowadays too, we're using the stronger antibiotics due to all the germs, especially bacteria, got stronger over 100 years as well too. And this is the reason why I've probably seen them from the GPs too. If you've seen a GP, they're reluctant to give you the antibiotics, even if you're dead serious to this ill, unless you're really ill too. Okay, or by most of the time they will say there could be viral infections too. That is something we're going to talk about later on too, how we're going to treat those infectious diseases too, how do we prevent them too. Yep.
If we have to talk about for example, boiling, do we have to know that it's 60 degrees?
Not specifically, but it's a common sense. Okay. I would say common sense because I have babies. Yeah, so it means that I need to UV sterilization, if you think about it, so there's the kind of things we're using that. If you haven't seen the blue light kill the germs, the bacteria, there's the things we're using a lot of times too. Not only in hospitals, probably in the kitchens a lot of too. Okay, but sanitations, using them dishwasher, they actually go up to 100 degrees Celsius, kill all the germs as well. Yep. Any questions about how they get adapted to? Out of the many topics here, I know these two, the infectious disease in agriculture and some of the adaptation is, I would say, probably the most boring topic out of the whole module 7. Koch and Pasteur experiment is quite interesting too. What we're going to learn next week in chapter 3 is all about this. We're going to learn about the response to the pathogen. This is going to be a little more interesting, except the plants. Go more depth into it. So what I recommend to preparing for, as you're having your half year is coming in a few weeks too, you want to get the module 7 contents. You're probably going to get like a module 7 contents in like week, next term. What I recommend to do is you make like a summary note to every dot points too, and try to understand, make like some details there too. Use my tables, use more some worksheet, and all the homework that I'm giving you too will be help you to like prepare for the exams coming. But the chance you get the adaptation, chance you get like some pathogen agriculture will be slightly lower. I would say 20 to 30% on these two. I'm more focused on the Koch and the Pasteur post-postulate first of all too. That's a priority for this chapter, and move it on to next. Any questions? Okay. So what I want you to do now is let's try some of the worksheet questions that's given. So it'll be great if you get started from the page 2, 3. Let's finish up with some of the Koch explained too, up to page, out of page 4, and if you have a chance, finish up to page 6, please. Let's start on page 2. This is a re-demonstrating the Pasteur's experiment, showing that flask sit stills open up, and there's a kind of silt going like kind of shifted a little bit. 1. Students set up the experiment to replicate Pasteur's experiment. Nutrients broth. So page 2, question 1. Was added to the 2 test tubes, and the tubes were sealed. Both tubes were heated for several minutes in the oven over a Bunsen burner. After cooling, tube 1 was uncovered and tube 2 was left covered. The student observations are described as that. So we're seeing the test tube 1 and 2 uncovered and covered. Observation is given to A. Did the students collect the qualitative and quantitative data? Now, the difference between qualitative and quantitative is something related to the measurements of the data too, with the numbers. Or like so if the student measured the volumes or the amount of the fungi, amount of bacteria, that would be quantitative. But from the result, he only observed that qualitatively. So seeing the measurement of whether they happen or not. Remember the difference between qualitative and quantitative, guys, please. B. Explain your reason for the answer in the 2A. 2A. Oh. So we have to start from 2. Tube A. Kind of lost the reason why the student results were different from the Pasteur result too. Oh, so it should be 1A. Why this must be qualitative? The student is a record of the records of the what actually record on observation. Okay, about how the growth, and there'll be no numerical. There should be 1A, so that's typo. Numerical, like the data were collected. There's a reason why this must be qualitative, not the quantitative given. 2. Suggest the reason why the students' results were different from the Pasteur's result. So let's go have a look at the results again too. We have uncovered and covered observation birth day 2. It both shows that observation birth and after 10 days, cloudy and cloudy. Why do you think we have that? According to the Pasteur germ theory, we kind of assume that student might not boil the set of the test tubes or like those tubes and it's sufficient enough too. So there's a chance that student did not did not boil the flask long enough or sufficient enough. And the purpose of the boiling is to what? To kill any microbes the already present in the broth. Present in the broth. And this is proven by, and this is the reason why, after observation, after 10 days, both of the covered and uncovered will have the same result, the cloudies too. And that shouldn't be, because if there is no microbes and there's no added microbes into the flask, there shouldn't be any bacterial growth. Okay, which was shown in the Pasteur's experiment, not on the student's experiment in the case of this. All right, let's move on to Koch, the experiment there too. Miasma theory proposed the noxious clouds of gas, also called the bad air or the night air, contain the diseases such as cholera and the black plague, and the people become sick by the breathing in the gas. The theory had been popular and was commonly believed for many centuries until Robert Koch work in the 1800s demonstrated that the microorganisms cause the disease. This finding became known as the germ theory, and Robert Koch work with the anthrax bacterium allowed him to propose the 4 basic criteria, which we can discuss that as Koch's postulate. Question 3. Carry out your own research and briefly outline how Louis Pasteur's discovery that heating kills microbes is used in the food manufacturing the process today's too. Give us some understanding about, this is basically about what? Known as the Pasteuralization that we discussed in the class today too. Let's be a little more specific about the pasteurizations. It's basically what we use in the foods, any foods that we have to use in the mass productions too, we use in this method. So this makes the food safe. So for example, let's talk about the water specifically too. Milk is heated to up to the 71.7 degrees Celsius or 72 degrees Celsius for really short period of time, 15 to 25 seconds. Okay, and this is basically how to kill the bacteria from that. Okay, and that's basically killing all that pathogens, harmful, the microbes in the raw milk. Another thing we use in that is if you ever received the dish in airplane, okay, if you ever realize that they are sealed and properly packed as well too. This is also time we use in the kind of some sort of the pasteurizations too. We put it into the really hot heat, like a high temperature for a really short period of time, so that it doesn't really change the taste of the food a lot, but still kill the harmful like the pathogens inside of that too, because that's really important in like especially the mass productions too. Okay, that's a good example. We can use it out. Question 4, carry out your own research and explain how contribution to Louis Pasteur and Robert Koch has contributed to our understanding of the microbiology and infectious diseases too. This is basically what I went through that today in your workbook to this too, in terms of that what they have done. Those two are very important scientists in not only for the science, in our everyday life. I mean, without them, you probably we're probably not here in terms of that. After that discovery of the microorganisms, the population of the humans, like it grows like it was exponentially increased too. Okay, so you can think about like how did they actually perform that too. Related to that, Pasteur is specifically related to swan neck plus the experiment, which is leading to the germ theory, and proved that spontaneous generation was incorrect. And then his work also encouraged the belief that microorganisms were in the air and could cause the disease. That's what we're understanding from that. Koch proved the germ theory through the experiment, and he proved that many of the diseases actually by the germs or the bacteria specifically too. Especially anthrax, cholera, or the TB is kind of too. So even with the tuberculosis, that's like vaccines we give it in the really young age there too, from the infants to the 1 year, all the diseases here. So they are both providing us the how to first prevent the infectious disease, two, how to cure them too. Kind of developing the ideas about microorganisms, microbiologies, and more and that too. Okay. Next page is basically talking about a little more the understanding about these two, so miasma theory and the germ theory. Let's talk about this more in depth. Miasma theory, also been known as the spontaneous growth. So in the miasma theory, what is the cause of disease based on the theory? It is basically the bad air. Bad air is the one there too. So as I said, this is the reason why the doctors use the word of kind of funny shape, kind of mask. They look like this. You have it in the horror movies too. Basically, they have something incense like over here too, so that they don't really inhale the bad air. Okay, so those are the actual doctors. They're wearing that too. But germ theory, we don't really think that. We think the microbes are causing the disease. When did this theory gain acceptance? Since ancient time. It's from the ancient time. And especially in the Middle Ages. Of the older countries too. But germ theory only be accepted from the mid-1800s after the contribution of the Pasteur and the Koch. Is this theory accepted now? No for the miasma theory, but we do, yes. And we are developing the theory even further more now too. Okay, don't worry too much about Jon Snow, or the Brosling. That's basically talking about different stories too. But if you're interested, you can watch the video of the TEDs. But if you don't have a time, don't worry about it. Okay, now the rest of those would be the kind of depth study kind of questions. You can try that too, including the questions at the back, especially about Koch's postulate and cause and effect of disease in agriculture too. Try these and try also your homework that I'm giving you out today too. This is basically like a depth study. I'm including some more detailed answers for the questions too. Use that as resources, and also try these homework. Homework questions could be a little challenging because it goes asking you some depth of the what we learned today, but it would be worth it to try these again too. There you go. The germ theory, all that will be given. So we're gonna start with a little like a short question next week. I'll ask you about the germ theory again, the contribution of the Koch and Pasteur's too. If you can memorize a couple of the some infectious disease in agriculture, that'd be great in terms of the factors all too. So we're gonna start from that and move it on to the how those we response to the pathogens. Start with the plant and move it on to the animals, specifically human immune systems, and that will be. Probably, I would say that'll be a highlight of these modules too. Our immune system, how they're working towards the pathogens, kind of reacting towards to it. Okay, any questions?